My name is Tara Ramirez. I'm a biology major at the University of Puerto Rico, Rio Piedras. This summer we were basically looking at how populations of neurons respond to visual stimuli. What we do is that we inject special fluorescent molecules into the mouse brain. That way we can label the particular populations of neurons that we're interested in looking at. And then we will take a microscopy and we will take the live mouse, expose it to some visual stimuli, and actually record the activity of those neurons live. We were looking at two populations of visual cortex neurons. One of the populations projects to the prefrontal cortex and sends information to higher brain areas, and then the other population doesn't project. And we were trying to look at whether there's differences in the properties of these neurons. What I've been doing mostly is just the data analysis. I do some image processing and I also do analysis on MATLAB. So our hypothesis was actually that those properties were going to be different because we expected that if this brain region is doing some very complex functions like guiding behavior, for example, that it might need some kind of specialized information. But our results actually suggest that it's just taking a broad range of visual information, so there's no difference between the two populations of neurons. It was interesting. I feel very odd at first about negative results. They're very interesting findings because it means that the visual cortex and the brain in general, it has very different ways of communicating. It's nice to know that in science, things don't always work out, but it's fine and you learn interesting stuff along the way anyway. So.